what I want to start with is that this work is largely, I'm going to report largely on things that were done by two people in my group, Mia Morel, who is now a graduate student at NYU, and Audrey Silverberg, who is now faculty member in Minnesota in the neuroscience department. And then some of these things, some of the more philosophical stuff that I'm going to be talking about, this has come out from many discussions with my friends and collaborators, David Schwab, Pankaj Mehta, Thierry Morin, and Philip Flake. And with that, let's go to this question, why should a physicist study life, right? In particular, brain. I spend a lot of my time on studying neuroscience, studying brain. And for me, sort of this question, this idea of maybe physicists can study something a bit more than condensed matter physics or cosmology or astrophysics, all of those things. It actually goes back all the way to when I was a kid, and I read this quote from Immanuel Kant that there's only two things in the world that fill my mind with an ever-increasing wonder and awe, and these are the infinite depths of the starry skies and the infinite depths of the moral law, right? And if you sort of squint hard enough, then what he's telling us that there's two things that are worth spending your time on, which is how did this whole universe start, right? The cosmology part, right? What it is that they're underneath, and then what is the consciousness? What is the brain? How does that emerge from the interactions of physical systems? And at some point later in my life, I had an argument with David Gross, who some of you know, what is more important or what's more interesting to study. Is it really traditional physics, string theory versus more biological things? And my argument to him was that studying string theory cannot be more exciting than studying the brain that can study string theory. And that's why I think that fundamentally this is a subject I want to spend my life on, and I continue to spend my life on. And so if we are studying life as physicists, which is very different than non-living systems, what it is that we expect to find. I am not in the mood nowadays to actually model some specific molecular circuits. Does this gene activate that particular gene, or does this neuron activate that particular neuron? I sort of understand that if we try hard enough, we will make it work, right? There is no new life force underneath there. Molecules are the same molecules whether they're alive or whether they're part of the living system or part of the non-living systems. We can make it work. We can make it a model of the underlying system. But what I am particularly excited to find, and I expect and I hope to find, as this new laws of physics, other laws of physics that Schrödinger has told us about, that of course living matter cannot elude the traditional laws of physics that we all know, but there should be something else beyond that, right? And I think that right now, in the last few years, we have started to see that there might actually be something else, some new laws that we can find if we just look hard enough, that we did not see in traditional physics. And so why are old laws not enough? I'm going to, this is an introduction. I'm going to keep on showing you beautiful quotes that I love. This quote specifically is on my office door and has been there for the whole decade that I've been here at Emory. This is a short story by Borges, and I'm going to try to read it for you, right? In that empire, the craft of cartography attained such perfection that the map of a single province covered the space of an entire city, and the map of an empire itself of an entire province. In the course of time, these extensive maps were found somehow wanting, and so the College of Cartographers evolved a map of the empire that was on the same scale as the empire and coincided with it point for point. Less attentive to the study of cartography, succeeding generations came to judge a map of such magnitude cumbersome, and not without irreverence, they abandoned it to the rigors of sun and rain. In the western deserts, scattered fragments of the map are still to be found, sheltering an occasional beast to beggar, and as a whole nation, no other relatives left of the discipline of geography. Okay? And I think that that's what's happening in our traditional sort of approaches to modeling, to understanding biology, is that we're moving along the direction of building these maps on the scales of 1-to-1, where we're trying to write a differential equation or a stochastic equation or whatever it is that we're using, but for every particular player, every molecule, every gene, every individual molecule inside a certain organism. And that might not quite work. And indeed, I mean, just this quote, this short story, you maybe want to compare it to the thing that we see nowadays in biology, so different atlases, like gene expression atlases in the brain, or different cell types, or different atlases of different organisms in population, and so on and so forth. This is precisely that map on the scale of 1-to-1 of what's actually going on in the population on individual organisms or population of neurons, one neuron at a time, or population of genes, one gene at a time, right? That's precisely the map that Borges is warning us about that maybe we should try to avoid. And so what's the problem with this old laws? Why shouldn't we be pushing it if we can model the whole organism on a scale of 1 to 1? Why shouldn't we be doing that? And I think that the point is that we can't, right? So the old laws, they work extremely well when you're dealing with a system that has a few relatively well-known interacting components. So these are equations from the famous Hodgkin-Huxley papers on modeling action potentials in neurons. And, these are clearly traditional ordinary differential equation that you would expect to see in some electrodynamics books, which this is what that paper is about, electric currents inside the neurons. And it works incredibly well if you can measure most of those parameters. But it doesn't, this type of approaches do not work well when the number of degrees of freedom that you are dealing with is in the millions, like in this picture to the left here, right? These components are not very well known. We don't know who talks to whom. We don't know what the various conductances are. In principle, we can try to go ahead and try to measure every single one of them, but then the sensitivities of this differential equation to small parameters, you pass over some bifurcation point, and that's it. You cannot really make any kind of prediction from just small errors that keep on accumulating, right? It gets even worse, right? You can try to write down the differential equations, for example, in population genetics or in ecological systems. For example, you can write the generalized local Altera models in those systems, but we, for bacteria, have even no idea what the degrees of freedom are. What is a species, right? We still have no idea how to account for that in the context of bacterial populations. And then, what are we talking about? How can we really take sort of this detailed microscopic descriptions to describe whole big communities if we don't know which components are the dynamical variables? And so yes, we probably want to, hope to find some kind of new laws. And why do we think that these new laws should be there? And again, it's not a surprise coming from statistical physics background that we expect the new laws to be there because more is different, right? And we clearly are dealing with systems which have a lot of components, and because of various generalized central limits theorems, because of locality, symmetry, in this type of systems, you will get effective universal descriptions. So at least the physicists in the hopes that for all of this biological system that we're talking about, there are going to be universal effective descriptions. And we just need to do, what we just need to do is to not model bulldozers with quarks. We shouldn't model the systems on a scale of an individual molecule if we're trying to understand what the brain does, right? But we need to find those descriptions on the right scale, which captures the phenomenon of interest and nothing else. And that's where the new laws will come. That's the laws that we've been looking at. And what I will try to show you today is at least two specific examples where maybe some hints of these universal laws are showing themselves if you squint hard enough and you really believe yourself, right? So when we're talking about biology being being different, it's pretty clear that in biology we really do have a lot of this more, right? So this is the two papers that are going to be the starting point of much of the presentation today. One is a paper by David Tang's group, and the other one is Anne Churchland's group. And so here we are in the first picture recording neural activity of about 1,000 hippocampal neurons. This is the mouse which is running on the virtual track. It moves, to the end of the track and then immediately transported virtually back to the beginning of the track. So it's a Zoom world, right? Back and forth, and you're recording from about 1,000 neurons in the hippocampus of this mouse, and you see the raster plot, this sort of cyan plot where dark points show up when a particular neuron is more likely to fire at a particular point in this on this track, right? So we have an activity of 1,000 and maybe in a few years 10,000 neurons that can be measured at any given point in time for a duration which is actually rather long. A mouse can be run on this track for tens, if not hundreds of times. So hundreds of repetitions of this particular experiment can be done. In the pictures below, these are This is sort of details of the experiment by Anne Churchland. So there, a mouse is trying to make a decision. Mouse hears a sequence of clicks. These are Poisson distributed clicks with a rate of either 10 or 20 hertz. And the mouse is supposed to make a decision, is it hearing 10 hertz on average, or is it hearing 20 hertz on average, right? And if it's 10, it should go to the left terminal and push the left button. If it's 20, it should go to the right and push the right button. And so here we are recording from a couple hundred, 3 to 4 or 5, whatever, depending on multiple different mice have slightly different numbers that are recorded from of neurons. And on top of that, we actually know whether the neurons that are being recorded from are excitatory or inhibitory neurons, right? The green ones as excitatory, the red ones as inhibitory, and the third panel. And I really wish I had a mouse here, but for some reason my mouse is not working right now. But the third panel, one second, could you please stay with me for just 1 second? I'll do another attempt. Yeah, whatever. I just don't have the mouse cursor right now. And so the sort of this panels is a superimposition of the excitatory and inhibitory neurons at the same time. And again, you have a raster plot on the right where each of the neurons is its activity shown with the green dot on a blue background. You have a beginning of the experiment, and at a certain point there is a tone which tells the animal to get ready. Then there is a stimulus, which is going to be a few clicks that is at 10 or 20 hertz on average. And then sometime later, a mouse has to make a choice, and then it makes the right choice and makes a reward. That's the scatter plot there. So you get this beautiful recordings. That's a lot, right? This is a lot of neurons at the same time. And what do we do with that afterwards? That's going to be the question that we'll keep talking about for the rest of the talk. But before I go there, I want to hammer a bit more on this idea that biology has more, right? Every field of biology that we're dealing with right now, the reason maybe that some of them are so interesting and so exciting nowadays is that everywhere we have more. So this is a paper from our friends taken in paper from 2010, where what's happening is every single B cell in blood of fish is being sequenced, and one can calculate the frequency of occurrence of a certain types of B cells in the whole blood of the animal, right? So we have the entire antibody repertoire of the animal known. And these are different animals, A through N are different animals for which this has been done, right? Another similar, for example, result from the sequencing domain, you can take an animal and take a whole body pathogen, in this case, HIV sequencing from this, and count how many times this or that particular sequence has appeared in an certain protein, right? This would be the GAG or an AF protein. So how many times does a particular sequence for this or that protein has shown up in that particular organism? And you clearly see some interesting patterns on both left and right. There is a standard 1/f zip plot for frequencies, both for the antibody diversity on the left and for the HIV sequencing on the right. That's some interesting universal feature that seems to be showing up both on the pathogen side and on the host side. And so maybe that's worth explaining. Maybe that's, again, more telling us that some universal features are going to show up. But very similar, actually, 1/f style plots also show up in behaviors that are not immune at all. Like, for example, animal collective behavior, flocking, and so on and so forth, have very similar statistics and similar large amounts of data, where we're recording from hundreds or thousands of individual components for hundreds of thousands of times, right? And so overall, whichever field you look at, whether that's gene expression or chromosomal accessibility or cell mechanics or microbiome composition or neuroscience or immunology, everywhere we have these huge data sets with the hints of some kind of universal patterns showing up. And the question is, what do we do with all of that, right? What do we actually do with these data sets? One possibility is something like that, where you can go to your, to Amazon, and you can see that a lot of people have just made a wall art out of that. And maybe that's actually a good use of this data, beautiful pictures of flocking birds and beautiful pictures of antibodies. But I would like to hope that there is something more there, right? That we can actually understand the underlying biology besides just printing it and putting it on the wall. And so why has it been difficult to get, the different, right? In the more is different phrase, we have more, can we get the different? Why has it been hard? Is that the reason is that traditional theoretical tools are usually, for extracting different, for extracting new emergent behaviors, are usually built on either symmetry, locality, homogeneity, and all of those features together, which clearly in biology at least do not obviously exist, right? The brain is not translationally invariant, brain is not symmetric, it maybe is not fully, randomly connected, but clearly it's also not totally local at all, because you can get axons that are connecting across all the brain lengths and so on and so forth, right? So in these cases, intuition fails. We just don't even know how the emergent theory is going to look like, right? We just don't know of examples of emergent theory in this non-symmetric, non-local, non-homogeneous situations, and we don't even know where to try. And that's what the biggest, hardest part, I think, has been. What's even maybe worse is that we have very little understanding of what is surprising and what is not surprising in such systems. So you go and you make some kind of recording. You see this 1/f plot that seems to be showing up in many, many, many different domains, like what I've shown you before. This is surprising. Should I have expected that? Is it a feature of a large data set, or is it the feature of the fact that the immune system is working very hard to do something, right? Should it be part of my theory, or will it just emerge from any theory for free, right? We don't understand any of this stuff as soon as we move from just a handful of degrees of freedom to 1,000 degrees of freedom, which are not symmetric, unlike an ideal gas where every molecule is about the same as every other molecule. And so, having thought about this for some years, my ideas for how to move forward in this particular in the direction of trying to see emergent effective degrees of freedom in biological systems is this realization that in a lot of situations, a lot of high throughput data sets are fitted explicitly well by models with random networks of interacting components. I think, for example, in ecology, a lot of work that was done by Boston University group by Pankaj Mehta and his colleagues shows that the bacterial community is very well approximated by random interaction methods, right? Not just purely random, but random with some kind of constraints, with some kind of symmetries on not necessarily individual interactions, but distributions of interactions, right? So with constraints coming from the general statistical biological knowledge of this system. And what I will try to do today is to show you a couple of examples in neuroscience where the same is true, where random networks seem to be explaining some of biological data. What it turns out that once you start throwing in random network models, there are a lot of universalities that show up essentially in the biological domain. And then we'll see this again. What also starts happening is that some of these random network models are equivalent to existence of relatively low dimensional latent models of the underlying data, and these latent features that describe the data quite well could easily be emergent degrees of freedom, or they could be imposed degrees of freedom. We don't quite know that, but the point is that they explain that the dimensionality of the model is much smaller than the dimensionality of the data. And while we don't really know how to extract these latent features and how to even choose what is the right ensemble of random matrices to describe a particular data set that we're dealing with, we're trying, as well as many others, even some people in this room right now, or this virtual room right now. And so this is my introduction, right? What I will want to show you, and maybe another slide, let's view it as an introduction. Not everybody in the room had actually seen random matrices, so I'm going to show you just one slide to show how beautiful universal features in random matrices actually emerge. So suppose this is a data that we we're trying to deal with. The typical data set in biology would be you're sampling from n units, these are neurons or presence of different mutations in particular organisms or whatever it is, right? Some features, right? 0, 1s, they're either there or not. And then you sample them a couple of times, a couple hundred, couple thousand times, right? So number of samples in this picture is 1,000, and there is 100 variables we're dealing with. And then the first thing that we're going to try to do, we're going to try to see how everybody is correlated with everybody else, right? So we're going to calculate maybe principal components for the covariance matrix of this data or something like that, right? And what you've all, or I think many of you, maybe not everybody, have seen is that if you take non-interacting variables, completely non-interacting variables, sample this, a couple thousand times, you calculate covariance matrix of those non-interacting variables, and you calculate the spectrum of the principal components of this covariance matrix, what you will see is something that looks like this. If I have 30 variables and 10 30 observations of 10 variables, if I have two different realizations of this data, there's no any kind of interesting universal behavior. There is, sort of 10 different eigenvalues in one hand, 10 different eigenvalues in the other example. They don't really look anything like similar to each other. If I have 100 variables measured as 300 times, and I calculate the covariance matrices and measure the eigenvalues, what you will see is the spectrum that is shown in the middle row. On the left is one realization of that, on the right is a second realization, and now the two start looking pretty close to each other, right? And if I have 1,000 variables measured 3,000 times, you can barely distinguish one realization of this ensemble from another realization of this ensemble. This is what's called the Marchenko-Pasteur spectrum. Right? That's just one example. And I think that what we will see is that very similar universality starts showing up and controlling dynamics and statistics of recordings from large neural data sets. Right? So that's my introduction. If you have questions, let me stop here for a few seconds, and then I'll move on to the meat of the talk. Okay.
I don't see any hands, so.
Good. So this is more details the experiment by Anne Churchland and her graduate students. Let's just go through this experiment again and try to explain what people have measured, what did they expect to measure, and so on and so forth. So there is a mouse. Mouse is hearing the clicks, and it either goes to the left spot or the right spot. And there is a standard the psychometric curve that the mouse makes errors when the 50-50 when it 15 clicks per second, and it's almost always right if it really is 10 clicks per second or 20 clicks per second on the other end, right? So we're recording from both excitatory neurons and inhibitory neurons, green and red, and the idea in the field has been that when the mouse makes a decision, there are some neurons which are accumulating the decision that we're having. We need to go to the left, 10 clicks per second versus going to the right, 20 clicks per second. And these neurons accumulate an evidence. And as the evidence for one choice increases, it's a winner take all. You excite some inhibitory neural pool. Those inhibitory neurons inhibit all excitatory neurons. And so the ones that actually had the evidence which is high are going to win. The animal is going to make a choice, right? That's the first picture, which is called here non-selective, meaning that in this case, it's only excitatory neurons that are selective that will be able to predict which way the animal is going to go. Why? Because all of the excitatory neurons are inhibiting the same inhibitory pool, and that inhibitory pool basically makes sure that everybody is inhibited except for one pool of neurons which are actually accumulating evidence for one particular outcome, right? So it's a winner-take-all situation. The role of inhibition is to make sure that there is one winner, and that's it. And so in this particular experiment, the experimentalists went on and measured how well they could predict which way the animal would go if they built a certain classifier based on activity of just excitatory neurons, just inhibitory neurons, and both recordings from excitatory and inhibitory neurons. And the big surprise is that the inhibitory cells we're almost as good of predictors of where the animal is going to go into the future, as the excitatory cells were. And so that means that this first picture with a single big inhibitory pool cannot explain this data, because those neurons are not selective. They are just activated to suppress everybody besides one winning group. And so what one needs is a more detailed, network of interactions where every excitatory neurons have their own private pool of inhibitory neurons. And so excitatory neurons get active, they activate their own private pool of inhibitory neurons. And then those neurons are going to inhibit everybody else, right? Both excitatory, everybody else, and inhibitory, everybody else. And so now there is activity in a single, pool of excitatory neurons and a single pool of inhibitory neurons. And so you can predict which way the animal is going to go equally well by recording from excitation, excitatory neurons or inhibitory neurons. So that's the idea. It's a very detailed network, explains the data quite well. And what we decided to ask instead, what would happen if I chose a random excitatory inhibitory network of neurons, balanced network of neurons, and let it run and record the activity in this random network and try to make a prediction which way the animal is going to go. This work was done with Audrey Sederberg. So we built a network of 100 or so neurons. 300, I think in the experiment there were 300 neurons. We built about the same. They're 80-20, meaning 80% excitatory neurons, 20% inhibitory neurons, which is typically what happens in different brain areas. They are coupled in terms of, with some kind of rate equations where, standard neural networks type equations where activity of one neuron is affected by the input from all other neurons, right? The network runs, it gets an input current, it runs, there is some activity that emerges, and then what we do is we train a linear classifier to predict whether the animal has been activated by, or the network has been activated by input type 1, 10 clicks, or input time 2, 20 clicks. And we can train a classifier only on excitatory neurons, only on inhibitory neurons, or on all of them, right? And so in the picture, I will always be showing, blue as excitatory, red as inhibitory, and black as both excitatory and inhibitory. There is no learning, no adaptation. It's just a plain vanilla random network of neurons talking to each other, and we're looking at how it responds to different types of stimuli. And that's what happens. So you can ask how many of the neurons in this network of about 300 neurons, which fraction of neurons, both excitatory or inhibitory, start responding selectively to just one type of stimulus, right? So connections are there, everybody is uniformly connected, but it's uniform randomly. There's going to be some fluctuation. Some neurons are more likely to be connected to some others with slightly stronger connection. And those activities are eventually going to get amplified by the dynamics of the network, right? And so what happens is, if you record from excitatory neurons, somewhere between 10 to 15% of them are preferentially responding to one stimulus and to another just by chance. And the same story is true for the inhibitory neurons as well. And here I'm showing in the experiments, depending on which animal they measure from, which neurons from which animal they measure from, et cetera, that fraction of selective neurons was between 10 and 16%, both for excitation and inhibition. Here we have about 10% as well, but remember, in a neural system, you only record from neurons that actually are activated, otherwise you don't even know that they exist, right? And so you would expect that the fraction of active neurons and therefore selective neurons in an experiment is going to be slightly higher than in our system, where some neurons just don't respond, right? And so they would be not recorded from in a typical experiment. So that's good already, right? That both excitation and inhibition show more or less the same selectivity to different input types, different stimuli types. We can go further. We can record this activity, and we can now train classifiers, simple linear classifiers, to predict whether this network was activated by stimulus number 1 versus stimulus number 2, and we can train classifiers from either excitatory, inhibitory, or all of neurons. And of course, all is going to do slightly better because it has both pools, just more input to it. but actually not much better. Both excitation and inhibition are essentially indistinguishable from each other. They're all within 75, 80 to 90% range in predicting what has activated this particular network. And in an experiment, the numbers are the same, anywhere between 70 to 85% accuracy from just training a classifier based on the activity of neuron or of neurons. So basically, what we're saying is that that this result and essentially every other experimental result reported in this paper, we could quantitatively, with an accuracy of at least 1 and sometimes 2 significant digits, explain by a model of simple excitatory inhibitory 80-20 balanced neural network, right? So it's a random network with constraints, completely explains the results of this particular experiment, and you don't need to do any kind of fine-tuning, developing a new specific model of the system, and so on and so forth. So the thoughts here.
Sorry, can I interrupt?
Yeah, of course.
Going back, so you showed these 2 models in the beginning, and you had the selective, so there's non-selective versus signal selective, and the network you set up, was it like either one of these, or
no? The network doesn't have either the E1 pool or the E2 pool or the I1 or I2. You just have, let's say, 80% of neurons which are excitatory, 20% neurons which are inhibitory. They randomly talk to each other, right, with the standard recurrent neural network structure. The input signals project randomly again into either excitatory or inhibitory, we tried both neurons in this network at random. And then what happens is some modes of collective activity of these networks get excited more than other modes, just by chance, right? And if you were to focus on them, you would be able to detect to predict or to post-dict in this case, which particular stimulus had actually activated that network. You do not need to have any structure. Basically, in the random networks, there is enough of detailed structure by chance, but the only thing that you need to do is to let it emerge in the process of dynamics and then hook onto it.
As a follow-up, to the question, the input is into random nodes, randomly chosen nodes.
In our case, so the picture, what we've done is there's input which is with a random weight matrix projecting onto excitatory neurons in this network.
And the readout?
And the readouts we train. So that's exactly what they've done in the experiments, where they look at the activity of the neurons and then they train a classifier based on the activity in order to, it's a supervised learner in order to predict which way the animal is going to go, left or right. We did exactly the same thing, exactly the same classifiers, right? And it turned out that the distribution of weights in this classifier that we built was also within two significant digits, the same as the distribution of weights that was measured in the experiment. And these are 2 hands.
Yeah.
Can I go ahead? Okay. Really interesting. I was wondering if how does the behavior of the network depends on like the degree of connectivity of the network? Like if you change the fraction or how many to how many other neurons these things are talking, does that change? And if somehow that, like if you're seeing the inputs with different frequencies, like I see the right one versus the left one more often, and I change my degree of connectivity, I can encode like a prior of what I expect the signal to be, kind of thing?
I think this is a very good question. I have some answers, but not all, right? Because at some point we need to actually develop a theory for this, and for now we only have simulation. So let me try to answer this, right? So what I'm showing is that what would be nice to do is to try to write down a theory of dynamics of these networks in sort of very similar to, let's say, Bob May's theory of linear ecologies, right? And the equations actually very similar to linear ecology equations as neural network interactions with some random weights, say mathematically indistinguishable. So we couldn't do that, but what we could do is we could do numerics. And what we see that we understand what actually happens in the dynamics, what it is that we actually latch on, right? So it turns out that there are some collective modes in this network that have large eigenvalues, so it's sort of the large real part and sometimes large imaginary part eigenvalues. These modes start growing very quickly, and these are precisely the modes which allow us to, which basically take small differences in the inputs and amplify them very strongly so that you can make reliable distinctions. And so to answer your question, I think what's going to happen is that as long as your network connectivity is sufficiently dense to generate a large number of collective modes which grow sufficiently quickly so that you can latch on them, then you would still be able to do the classification, right? The details of what the connectivity is, et cetera, will probably matter a lot less than the details of the structure of the eigenvalues of the linearized version of this network. And then the second question, whether you can impose any kind of priors, et cetera, I haven't thought about this at all, so I just don't know.
Yeah, we have, I don't know if we have a question from Milo, maybe from Michael too, I'm not sure.
Yeah, I had a question about how important is it that it's a linear model?
So we started with a model being nonlinear, right? It's only in order to study how the model behaves, the network is sort of in steady state, nothing is happening, then you run it, it starts activating. Eventually, nonlinearity starts mattering, but which particular collective features are going to grow, which particular eigen vectors are going to grow, that you can study in a linear regime, right? And that's essentially the same style analysis that Bob made it in 72.
I see. Okay, so you're doing like a linear response kind of theory on a fixed point.
Yeah, around the fixed point of no input dynamics.
Cool.
So just to finish this off, and I will move to the second part, right? We can do a lot of other interesting things, but the point is that this emergent selectivity is basically, it's equivalent in this case to emergent latent distributed dynamics. There are some collective degrees of freedom that suddenly show up and start running and start activating themselves and start developing that would not be obvious from activity of individual units, right? Is brain really random? Probably not, right? This model is incredibly simplistic, right? But it's interesting to see how far you can push this idea of random with constraints, in this case, constraint being excitation inhibition balance network. How far can you push this network? And maybe a random network model, rather than sort of a detailed model with a bunch of pools of neurons talking to each other, should be the null model from which we should build, add details, rather than, a detailed model to which we add even more details, should be the null model from which we should start from. So that's sort of what I wanted to point here, right? That random networks work. Brain doesn't have to be random, but it's a good enough model for this type of experiments. And I think that an interesting question is that it seems that the experiments that we're doing are just not able to distinguish whether the underlying brain structure is random or not, right? And maybe we should be doing different experiments rather than the experiment that people are doing nowadays. So moving to the next part of the talk. So now that we understood that there are emergent effective degrees of freedom in the network, can we try to somehow figure out what they are, detect them from data? And a different group of people, David Tang did the experiment, and his graduate students did experiments, and then Linoy Mishulam, who was working with Bill Bialik, did the analysis. They sort of, in a very different experimental setup, they tried to ask this question: how do we get out the effective emergent collective degrees of freedom from the data, if those exist, right? So let's walk through their paper and then see how we can model it with random networks as well. So if there are emergent degrees of freedom, then effectively the only approach we know how to detect them coming from statistical physics is renormalization rule. And we're going to do some version of real space renormalization group. And that is, we're going to coarse grain neurons into some hyper neurons, big neurons, whatever we're going to call them, right? Super neurons, and see what happens to various interactions, various correlation functions in the system under this renormalization transformation. It's very difficult to do. It's impossible to do RG, classical, traditional RG in a network with in a system with no locality, right? With no space, because you don't know who neighbors are. And so what they've decided to do, which I think is the most important sort of realization of this paper by Illinois, is that you can replace notion of locality by correlation, right? Two units that are the most correlated with each other, we're going to call them the most local, and we're going to combine the two of them together. And so instead of doing block spinning, what we're going to try to do is take neural traces, see the two neurons that are the most correlated, combine them together, just add, the activity of one is added to the activity of the other, then look at the next most correlated pair and add them to make the largest neuron, and so that the larger neuron, and this way you're doing hierarchical clustering, hierarchical cross-graining of the system, and you can calculate correlations between neurons, autocorrelations, temporal, spatial, as a function of how many coarsening steps you have done, okay? That's the idea. Yes, there's a question.
Yeah, so in terms of this part of the modeling where you're trying to use correlation as a measure for kind of connectedness, do you guys use the direct correlation to do the mapping, or do you have like some kind of underlying, let's say, spin-like model of direct couplings that give rise to, because a lot of correlations can be through indirect, like A is correlated to B because C is connected both of them. So are you first mapping to an underlying direct interaction model to build these distances, or are you using just the correlations directly to build this model?
I mean, this is a great question, and that's sort of reducible versus irreducible discussion is always permeating this type of analysis. So here, first of all, you cannot really build an Ising model in 1,000 neurons, or rather infer Ising model in 1,000 neurons, just too many neurons, right? One cannot invert the covariant matrix and actually get the actual interaction. So here we're working on total correlations. But then the idea is that the strongest neurons cannot really be, the strongest couplings cannot really be explained by a confluence of many, many other couplets, right? And we're really here focusing on just the first few steps of coarse graining, where we're taking the largest correlations. We don't care about the smallest one, which could be induced, right? We care about really the largest ones. And so there is a chance, like my bet is that most of the correlations that you're going to get this way are actually irreducible. Okay?
Thank you.
I think it's a great question, right? So what did they find? So as you increase, it will make larger and larger clusters of neurons. The temporal activity of this, what do we expect to find, right? That correlations between clusters at larger scale should start disappearing. Temporal activity of this large random clusters should decohere faster. Clusters, if they are sums of individual neurons, and if neurons are independent from each other, then the activity variance of this cluster should grow linearly with the cluster size if it's law of large numbers, sort of adding independent components, right? However, if all neurons are perfectly correlated with each other, then the variance of activity of this cluster should grow quadratically with the size, right? And so in reality, it'll probably be somewhere in the middle between linear and quadratic. And then the distribution of cluster activity, being that each cluster is a sum of many neurons, should approach the Gaussian, right? Central limit theorem. That's what one would expect. What they found is that the cluster activity shows power law distribution and correlations, both spatial and temporal. There are non-Gaussian distribution of large cluster activities. And what it really means that there is some kind of fine-tuning going on, probably, right? Because otherwise, how would you get sort of critical signatures in a system without fine-tuning, right? It's very difficult to do in traditional physical systems. So what we decided to do is to model this as a random network. And since we know that in a lot of random networks, you get this sort of emergence of latent features, all right? what we're going to try to do is a really simplistic network where you have a bunch of latent features, the green ones over here. Each one undergoes, some kind of random walk Langevin dynamics, and then all neurons are randomly connected to these random features, and that's it, right? It's a restricted Boltzmann machine style structure where each of the latent fields interacts, moves on its own, and all of the neurons are just coupled to the latent fields. That's it. Because we're recording from hippocampus has both what's called place cells and non-place cells, about 50-50 of them. We're going to also introduce here that some of the latent features actually are positions on the track. So they will account for spatial memory. Some are not. Connectivity in experiments here is about 50%. We're also going to do the same, sort of all the usual things that I've been telling you, that it's random with biologically realistic constraints, right? But beyond that, no adaptation, no fine-tuning of any kind. So then we do exactly the same procedure as Michel and all do. And what we see, for example, on the first plot, the activity variance, which I told you should be 1 if all neurons are independent from each other, or 2 if they're all dependent, shows a beautiful power law with a slope of about 1.367. And in an experiment, they got a plot slope of 1.4, right? The blue signatures. You can look at the scaling of the free energy. We got it from this model, which is basically a probability of neurons not being active at all, depending on the cluster size. And we got power law with a scale of 841. Experiments got 8.8. You can look at the distribution of eigenvalues of activities of these big clusters. We've got something which is like 0.65 plus minus 0.09. In the experiment, they get the same thing that is basically indistinguishable from what we have measured, right? So every single measurement that has been that they have calculated, we got exactly the same critical exponents with numerical accuracy of about 2 significant units. Madhav, I'm going to take your question in a minute. Let me just show you a few more things, okay, before we go there. So they've done it on multiple different mice. And so there are 3 different mice, and you can look at this different type of critical exponents, the spatial correlations, the temporal correlation, the variance exponents. The 3 different mice are shown by 3 different crosses. And in our model, by changing parameters of the models, we can get critical exponents, which are essentially the same, expanding the whole range of what real animals do. And the most interesting part that I mentioned to you is you would expect that the probability distribution of this big cluster of neurons should become Gaussian, right? In an experiment, the blue picture here is experiment. It doesn't. The activity, normalized activity of big clusters as you go to larger and larger coarse graining seems to have to saturate at something that is manifestly non-Gaussian, non-parabola on this log linear plot. And on the right is our model, and I'll let you see that the 2 are not just qualitatively but quantitatively the same. You get the same feature on both the left of the parabola side of the parabola and the right side of the parabola. And this is again without fine-tuning, just throwing the bipartite network of interactions onto each other. Now I think it's a good time for your question.
And I just had a question about both the paper from Bill, Michel et al. And your approach. They also suggest a second approach, which is sort of the analogy to momentum space.
Yeah.
And I guess even in that paper, they were at least maybe in my reading, they were relatively agnostic. I mean, in the physical case, of course, real space and momentum space equivalent, but they were kind of agnostic, or didn't really say what different things does one learn. Do you have a view on this, or did you pursue the momentum-based one?
This is momentum-based. So what's happening here, I'm a bit playing fast and loose here, but what's really happening here is the following. You get overall activity of your system, and then what you do is take principal components of this activity, and then you choose a subspace in the principal component space of n over 16, n over 32, n over 64, etc., etc. Neurons to decrease the number of neurons, and then you project the overall activity on that subspace, right? So principal components are effectively the same as momentum space for a system which is translation invariant. Principal components are the momentum space, right? So that's precisely what they did. It's precisely what we do here. The crucial part is, of course, whether you're doing in real or momentum, it's just linear transformation. So whatever is Gaussian in one is going to remain Gaussian in the other, to a large extent, not perfectly, but to a large extent. There are some other features there. And I think that the most important part is that this way you clearly observe that it's not a feature of which neurons you are combining with each other, but the large scale, where the scale is determined by the, how much variance of the overall activity you are explaining. Large scale features in the system are non-Gaussian. Even when you are adding a large number of neurons together, and on top of that, there clearly exists a large and large scale limit, right? You look at these pictures, n over 16, the probability distribution for coarse graining by 16 is of activities after coarse graining by a factor of 16 is quite different from that at 32, quite different from 32, but by the time you're getting at 128, they're all on top of each other. So there is hopefully a limit. You have gone, you basically got to the fixed point in your RG flow, right? And that fixed point is non-Gaussian. And that's a pretty cool result, right? That even if you keep on coarse graining further, you are not going to get to a Gaussian, which is interesting. Okay, so what I want to point out is that very similar model with latent features and external activity also explains, and I'm not going to explain how, it also explains the 1 over f scaling in the frequency distribution of antibodies, in the distribution of different protein fragments in the HIV. It also explains correlation functions in the bird flocks. And this is a part of the work we're still working on, because Mia has left, she was an undergraduate here doing this work, and now she's a graduate student working hard elsewhere. It also explains the emergence of avalanches in cortical circuits, right? You can view avalanches as really fine-tuning of excitation and inhibition, or you can view avalanches as being governed by some kind of latent features dynamics. And so the thoughts here that I want to end you, I want to sort of leave you with, is that none of the current experiments can distinguish these sort of regular models versus random models. And our intuition completely fails, right? But we would not have expected that a random model I would not have expected that a random model in both examples, in the case of EI network, can explain the complicated signatures in neural data, and the random bipartite model can explain this beautiful scalings in the hippocampal data. Like, I didn't expect that. I don't think anybody did, right? And that's the emergence, right? This is a part that we cannot really predict what's going to happen when you get many things together. Is brain really random? No, it's not, but it's probably a good model to start from. Maybe that should be the null model for our understanding of the experiment. So should we take these latent variables seriously? If yes, then what are these latent variables? One possibility is that the latent variables are basically stimuli, the external stimuli, which then just govern the activity of the neurons that are responding to them, right? So you have the latent features and the variables that are flickering in response to them. But then why is the brain so crazily complex if we can model it as basically every neuron being independent of every other neuron, conditional on those latent features? So maybe a better structure, maybe a better understanding is that these latent features are really not the external stimuli, but they are emergent latent dynamics, and the connectivity between the neurons is what makes the system look like latent dynamical variables, and then they govern conditionally independent neurons. And then can we understand when this, and under which conditions would this happen? I don't know yet. That's what we're working on, right? whether the latent features are external or emergent, what I want to stress is that how accurate these models are in explaining the statistics of the data. We're getting 2 significant digits from a random model, sometimes better than a fully tuned model designed specifically to predict these features. And so what I would hope is that this way, if we continue to trying to develop this latent emergent models of brain activity, maybe we will end up with a theory of the brain that doesn't have neurons, right? Just like we can maybe develop a theory of evolution which doesn't have genomes, because genome is, yes, it's an individual component, but on which selection is happening, this is, yes, the degree of freedom, but other collective units that move together, right? And then the question is, how do we find them, right? And here is a shout out to a couple of our colleagues here, Warden Berman, who is trying to understand, to extract these latent features in another very complicated system, namely behavior of the animals, and Jason Pandarinas, who is trying to extract these latent features in motor control in different animals. And I think that their methods are some of the best ones for extracting the latent features. And then we're meeting them on the other end, trying to explain that this latent features should always exist, right? And maybe at some point we will meet and we will then have not just the suggestion that theories should exist, but also a way of actually writing them down, okay? And so at this point, questions.
I can maybe start with questions. I was wondering if you have a sense of like why random networks have features in general. Like, it just seems like there's always something when I run a random model, something I input about the distribution that I could see in the measured thing. So like, is there some feature of like the input that you think is really kind of driving whatever you're measuring in the output?
I'm not sure I understand the question correctly, so let me try to answer what I think you're asking, right? And that's stop it if that's not the case, right? I think that one reason why random networks seem to work is because we're measuring wrong things, right? Because we're measuring things that do not differentiate between an activity of a random network and an activity of a fine-tuned, precisely developed network. And one way of how this type of approaches could be used is to really design better experiments. So for example, for the decision tasks, what one should be measuring is not just activities of individual neurons, but correlated activities between E and I neurons, or even higher order correlations, EEI, right? And if you can measure those, you would be able to distinguish whether this is a random network versus a non-random network. It's hard to do, but it's possible. The other viewpoint could be that the reason that they work well is because they really represent the underlying biology, and that would be my guess, right? And the reason is the following: that if you have a brain with 100 billion neurons, this brain needs to be trained to do certain tasks. You need to learn how to control your own muscles, your own things, in order to actually function in the real world, right? You do not have an unlimited amount of time to tweak every knob, as you know, in your control strategy when you're trying to move around the world, right? To tweak every knob and learn a particular control strategy for your own muscles, if that strategy is really cannot be summarized in terms of a small number of effective degrees of freedom. You just don't have the time. And every brain is different. We have only 20,000 genes. You cannot encode activity, or you cannot encode the detailed structure of a brain in 20,000 genes. What you can do is you can encode general properties of the connectivity, right? It's going to be a random network with constraints. And then the hope is that random network with constraints fall in a sort of almost automatically produce to you some kind of lower dimensional dynamics compared to what they could have. And then you are able to learn how to control that dynamics precisely because it's low dimensional, right? And your evolution is driving you to this low dimensionality because otherwise you cannot control your own body, right? Over an individual's lifetime. Does it make sense what I'm saying?
Yeah, I think I think it's, you're highlighting a little bit about the constraints being like present, and we can learn about those constraints maybe, but the experiments aren't necessarily designed to measure those constraints.
Yes. Yeah.
That's interesting. Thank you. It was a beautiful talk. Manuel has a question.
Yeah. Thank you. Great talk. This is really, really cool. This is the kind of question that made me look really stupid for asking things that I don't fully understand. But I couldn't stop thinking about Carl Friston's free energy principle related to your idea where, he claims that every system that self-organizes like a sentient being, something that is gonna sense the environment and respond to it, is gonna self-organize into this set of internal states and a Markov blanket that surrounds it, and make Bayesian inference on the inputs of what the environment is sending. So I was wondering if, in this system, you can look into whether some of the neurons of your random network act as like this just like the sensing neurons that then maybe that they're like isolating a group of neurons inside that represent internal state of what the mouse or the simulated organism is going to do. So you'll have like two clusters of cells. Like if this one's all turn on, is going to go to the left. If this one's all turn on, it's going to go to the right. While some of the other neurons are just acting as like the signal transduction system. Like they just like are the ones that receive the input, feed it into the internal system, and then that activates either of the two clusters. I don't know if my question makes sense, but
I don't think I know a lot about Friston's free energy principle to comment very intelligently, right? I'll say one thing that I think is potentially useful. So, of course, I've swept a lot of things under the rug in this case. I want to go to this slide. Okay. So under which conditions will all of the scaling and all that emerge in this system? So what's required is that the neurons are actually responsive to the latent variables. So the sensitivity between the green and the blue layer has to be of order 1, and then you are guaranteed that you will get all of the scaling laws that I'm talking about. And that's if you were to look at the green variables not as emergent latent features, but as let's say the stimulus that we're not observing, is an animal, how hungry it is, or what it's really seeing in the outside world, or whatever it is. And essentially, what the statement that I made about susceptibilities means is that the neurons have to be adapted to be responding to this latent features. They cannot be always on or always off. They have to go through their entire dynamic range in the course of the experiment. And when that happens, that's when you will start seeing the interesting scaling properties. And so the statement, maybe it's not about the free energy principle, because again, I don't know much there and I don't want to say something completely wrong, but I think the statement is that when you do not have useless neurons, when the neurons are all trying to contribute to the activities or get their activity in the Goldilocks range where they're never always active or never always passive, you will get this type of behavior. And I don't know how exactly it relates, but I think that there is some relation to what you are saying.
Yeah, great, thank you.
Question, can I ask?
Yeah. Whoever it is.
Yes. Yeah. So, like, can actual network be non-random in the following sense? So, what we want to have in our system is availability, right? So we expect that like during evolution, you don't break everything and don't build everything from scratch every time. So there should be kind of like some kind of path in the like network space that goes from like one stable point to another stable point. And if a network is like very special, what would it mean? It would mean that like all networks are the same, because otherwise, like, how can you get a transition from one very special case to another very special case during evolution? Does it make any sense?
I mean, I think that again, I do not think that we are talking about the same network, right? So what I'm talking about is an effective model of the underlying activity of the system. And what I'm saying is that the effective model is basically random. It doesn't mean that the actual structure there has to be. Okay? That's about as much as I possibly will say. And also, let's add another thing here. You can also train this network. So you the fact that it's random, but there are coupling constants, and they also can change in the process of learning. So, but so I'm not doing a good job answering the question because I didn't quite understand it. But I'm going to turn back to you and to Madhav. So you had this pretty cool paper not long ago on the shapes of wings, right? There's also wings, and one of the things that I was thinking about reading it, and it might be that it's just because if you have a hammer, everything looks like a nail, or maybe not, right? Is that the fact that you realize that there are only a couple of modes over which the wings actually change, and all of the mutations result in just a handful of large geometric pattern changes in the wing, is effectively a phenomenon of the same type that I'm saying here, that there are a handful of latent features in the geometric space. How they emerge from the true interaction genomic network, I have no idea, but they did, right? And so there are these latent features, and the shape of the wing can be produced by sort of combination of a handful of latent modes, right? Of principal component modes, right? So I think that what you are measuring is more or less of the same style, whether it's just an observation or whether it's more than observation can be made sort of more precise, I don't know. Right, and I think that definitely in your case, I mean, the network of development is not random, but it results in latent features. And the mapping between the latent features and the observable, latent variables and the observable features is pretty much random. You don't know which one goes to where, right? How particular gene affects what. You don't know that. So the effective network is essentially bipartite and random, while the true network is not.
Okay, yeah, thank you.
Go for it.
Okay. Can I ask another one?
Yeah.
Yeah.
Sorry. This one has to do, I guess, with this idea of emergence. You know, although I'm a big fan of more is different, I never understood if what Phil Anderson meant was something like, if you knew all of the microscopics, you could reconstruct the macroscopic behavior, but you don't need to. Or literally, even if you knew the microscopics and you have, you know, enough computational power, like you were like, I don't know, the Laplace's demon, you wouldn't get the emergent behavior. And you know, in the reason I'm asking is, like, in your models, you do know the microscopics of everything. So is there some metric, like, I don't know, some mutual information related stuff where you say, look, I don't gain information from knowing the microscopics because the macroscopic behavior gives me everything I need to know. So it's, you know, my personal not understanding what really more is different is trying to say.
I don't know what Phil Anderson meant, right? The only thing that I know is a paper that I read, and that's it, right? But my understanding of it is that, you know, because of, let's say, there's something that's unpredictable, right? You have chaos, you have positive Lyapunov exponents in your even classical dynamics, and so some of the things that you would, even if you had the exact equations, an exact million of equations, but you didn't know some parameter to 1% somewhere, as long as there is one Lyapunov exponent that is positive in that system, you wait long enough and you cannot make any predictions, right? So it's not that you just don't have things to a good enough accuracy, it makes no sense to measure them to even better accuracy, right? And here, it's not mutual information. We don't need to do that. It's like, I'm stuck at this plot, for example, just, you know, in some experiment, they measure alpha of 1 point, like the whole characterization of an experiment was a handful of critical exponents, alpha, beta, and mu. That's what they use to, and then there's another, the temporal exponent, z. So 4 exponents that they measured to characterize every mouse, right? And so here is their number 1.4, and ours is 1367, right? There is 0.88, ours is 0.84 with an error bar, right? So essentially, this model has explained everything that has been reported, that was previously had been reported by the experimentalists. I do not need a finer model, more detailed model to explain everything that had been reported by experimentalists. And I'm always a fan of when developing models of two things, right? If you are developing a model you develop it to explain a certain specific phenomena, because there doesn't exist a theory of everything, right? There is a theory for one particular thing that you are developing it for a handful of things, right? But then those things that you chose for your theory to explain, it needs to explain to the same accuracy as the experiments. You cannot just say, oh, it looks the same. No, it has to be the same numbers, right? So your theory has a finite domain of applicability, but once you've put out that domain, then within that domain, it should be quantitatively precise. Okay? And so that's what this data shows, right? I mean, clearly, I will not be able to reproduce the entire statistics of the firing of a mouse, because, I mean, this is not a mouse, this is just a simple bipartite network. But for calculating these critical exponents, nobody can do any better than we have, because we've done perfectly, right? So it's as good theory as any other would be.
I see, yeah. I guess, you know, my misunderstanding was something like emergent. The way I was thinking is, instead of having to solve, you know, 10 to the 23 differential equations for each molecule, I do a PDE and I get Navier-Stokes. So that's my effective theory.
Yeah, that's perfect. That's exactly, I think, correct, right?
Yeah, but in this case, you are modeling the microscopics. No, like it's not like you have like a single
That's
PDE.
absolutely correct, right? So we've only gone the first step. We have noticed that once we are modeling the microscopic structure, let's say in the first paper, in the first part of this presentation, we're modeling the entire hundreds of neurons, and suddenly we see that it's only the activity along a handful of collective dimensions that matter, and maybe those are the things that are the correct degrees of freedom at the higher scale.
Mm. Yeah, yeah.
We don't know yet, right? I mean, correct is not even a correct word, right? I mean, useful, it would be a better word. And whether useful or not means, you know, we need to do a bit more work. Right?
I see. Great, thank you.
Maybe we'll, should we switch to more open-ended questions or philosophical questions maybe?
This wasn't philosophical enough.
Yeah, I know. We haven't really stopped, but I guess people can ask about, you know, Ilya, his life, things, and thoughts on biophysics in general. I'll stop the recording.